Hello, I'm Kevin Davis with BioIT World with some commentary on a bit of breaking news that we have on our website, bioitworld.com, about Ion Torrent systems. Ion Torrent is one of the new generation, the third generation of DNA sequencing companies that we suspect are about to break out and reveal their technologies to the community, which is not before time because they've been in stealth mode for the past 3 years, and like many other companies in the space, many people are very interested in what they're cooking up. It's likely that we'll know much more by the end of February when their founder, Jonathan Rothberg, gives the closing talk at the Advances in Genome Biology and Technology Conference in Marco Island, Florida. That's a very coveted slot and a very important meeting, and we'll know much more then. But earlier this month, Ron Davis, Stanford professor, gave a talk at the Molecular Medicine Tri-Conference. That's the flagship conference of our parent company, CHI. And he presented a sneak peek at some of the technology that is likely to feature in the Ion Torrent platform. Ron Davis is an advisor to the company. So before we get into who Ion Torrent is and why you should care, let's just recap a little bit about how far next generation sequencing has come in the past few years. It was in 2005 that Rothberg's former company, 454 Life Sciences, company he founded, debuted the first commercial next-generation sequencing instrument, a very powerful instrument that could sequence about 100 million bases of DNA in a single day, which is a vast improvement on the traditional Sanger machines that have ruled for the previous 15, 20 years. Flash forward to today, and the Illumina and Life Technologies instruments in particular, the latest versions of those machines it is claimed, can sequence something like 200 to 300 billion bases in a single run. A run takes just over a week. And so if we compare notes from 2007 when 454 announced the sequence of the first human genome using next-generation sequencing methods, Jim Watson, to today, we've seen about 100-fold increase in throughput and about 100-fold reduction in price in just the space of about 3 years, which is an absolutely phenomenal rate of growth in performance and something that really leaves Moore's law in the dust. So that's the situation as far as second-generation sequencing methods is concerned, but there's enormous anticipation for what the next wave of technologies will bring, so-called third-generation machines. I'm not sure there's an agreed definition on third generation. Some people might focus on using single molecules, although the Helicos platform already uses single molecule DNA sequencing. Some might focus on a whole different range of technologies that more directly interrogate the DNA without having to resort to fancy confocal microscopes and lasers and all the rest of it. So there's a lot of interest in building these new technologies, and I think we're going to see the first of these appear in 2010 from companies like Pacific Biosciences and probably many more. Ion Torrent then has been in stealth mode for the past 3 years, but the reason I think it's a company that is worth including in the third generation discussion, the first reason is Rothberg himself. Underneath the boyish enthusiasm and the uncombed hair and the tendency to wear Mickey Mouse socks is a formidable scientist and businessman with a phenomenal track record. In the past 2 decades, Rothberg has founded Curagen, which he took public, then span off 454, as I mentioned, which has already made its mark in the sequencing field. And one of his more recent launches, very successfully in its own right, is Raindance Technologies. We've not been able to learn very much about Ion Torrent other than details that were released in a patent last year. But as I mentioned at the beginning, Ron Davis from Stanford, an advisor to the company, presented a little bit of information that I thought was worth sharing on some of the technology that his lab has licensed to Ion Torrent. This technology has been developed really for many years, and the key paper was published back in 2006. The chief architect and lead author was Nader Pourmond, a Iranian Swedish American scientist who has since now, although he still has a small footprint at Stanford, and now has his own group. He's on the faculty at the University of California, Santa Cruz. So we have a schematic taken from the 2006 paper published in the Proceedings of the National Academy of Sciences from Pourmond, Ron Davis and colleagues. And this is the basic setup. And it's really incredibly simple. We have an oligonucleotide attached to a solid metal electrode. In this case, it was gold. And as a nucleotide is brought in to be able to bind to its complementary base on the second strand of DNA, there's obviously chemical changes, but there are also electrical changes. And the one that Pourmond's group have focused on is an electrical change whereby, with the release of a proton that diffuses away into solution, there's an introduction of a net single negative charge into the DNA strand for every nucleotide that is incorporated, and you can go to the paper and see more details about this. With that charge, since the DNA is sitting on top of this polarized electrode, there's an induction of a compensating charge, and it results in a small transient current, which can obviously be detected. So in principle, if you repeat this process and cycle it and flow in different nucleotides and can reproducibly detect these transient currents, then you have the basis for a very straightforward, very first-order detection system. Now, Pourmond's group have published follow-up studies to this. In 2008, they published another paper showing the first demonstration of a grid of electrodes, so multiplexing this. I wish I could tell you where this stands now and be able to report on the success of actually seeing raw DNA sequence traces. We haven't seen any of those, to my knowledge. I haven't, and I don't believe any of those data have been published so far. Suffice it to say that Pourmond, as we quote in the story on bioitworld.com, says that it's going really, really well. So this is the basis of the technology that has been licensed to Ion Torrent, and there are certainly allusions to it in the patent that was granted to Rothberg last year. It's an exhaustive document, and I wouldn't claim to have read it all, but certainly that document talks about other ways that one might sense or detect the incorporation of nucleotides. Certainly there are chemical as well as electrical methods that you can potentially look for. You could look for changes in pH or the derivation of pyrophosphate byproducts or other chemical means. The full details, I'm sure, will play out soon enough. So, as I mentioned, at the end of February, I suspect we'll be hearing a lot more about what Ion Torrent has been cooking up for the past 3 years. The goal, at least according to Ron Davis, is to make not necessarily a massive throughput sequencer, but a much more portable sequencer. He talked about dreaming of having a handheld $10 sequencer that could really be taken into the field and would allow tremendous flexibility in allowing all kinds of experiments to be done without having to wait until you've got a human genome to sequence or multiple samples in order to make the expense of a run worthwhile. So that's where we'll leave it for now. We certainly look forward to the debut of Ion Torrent and many other companies in the third generation sequencing space, and we look forward to commenting on them using this media in the future. I'm Kevin Davis for BioIT World. Thanks for watching.